Hi everyone, today's talk is going to be a brief discussion about causal inference on aggregate neural signals. So as a brief overview of what I'm going to talk about, firstly, why would we worry about looking at causality in the brain? And then, what is the ubiquitous confounder? Basically, what impedes our ability to perform causality on the human brain? Thirdly, I want to discuss the homogeneity assumption and whether this is an impediment for causal inference. And fourthly, I'd like to just go through a possible framework for performing causal inference on aggregate neural signals, and I'll conclude by saying whether or not I think this is a thing to do. So why should we worry about causality? Personally, as a neuroscientist, I want to know about biological mechanisms. This requires some idea of causal relationships. I'm showing here the classic neuron synapse configuration, where an action potential generated in the blue cell, number 1, goes along the axon, releases neurotransmitter at the synapse, induces a change in the postsynaptic potential of cell 2, and when integrated with other signals, can produce a spike or an action potential. So this is the basic causal framework that we all understand as neuroscientists. But can we go further to actually infer causality in the entire brain? We can conceivably use what are called causal inference graphs to do this, or so-called do calculus. Along with some form of intervention or simulated intervention. I'm showing here a sort of notation we could use for this. So the red lightning bolt here with zeta beside it basically indicates that we are changing the activity of cell 1 to a value of zeta. So on the left of this little inequality, we're looking at the probability of activity in cell 2 at some point a little bit beyond t, given that we perform this action of setting the activity of x1 to zeta. We want to show that the activity of cell 2 is greater than what we would expect if we didn't perform this intervention. So that's what's shown on the right, and it's often referred to as a counterfactual. So this is a basic framework we could possibly use for inferring causality in the brain. So we know about neurons and synapses. However, measuring all neurons and synapses and their synaptic connections is a huge and I would say intractable task. So there are about 10 to the 11 neurons in the human brain, and each of those has between 10 to the 3 to 10 to the 5 synapses. This is a huge number of interacting components, and to make a causal graph this large would be basically impossible to develop causal inference on. So we can't model every single neuron in the brain. However, if we only measure some neurons, we have another problem. It means that we have some signals that are possible confounders which we have not recorded or measured. Maximally, this is the number of neurons in the brain. In practice, it's a lot less, but it's still a huge number. And these graphs are just sort of indicating how poorly this sort of framework scales. So we have a network of 20 nodes on the left, which is fairly tractable. When you scale that up by 2 orders of magnitude, it's already a large graph. Trying to put that to 10 to the 11 makes it basically impossible. So this is a problem. How do we address it? One thing we can do is think about aggregate signals. So our inference, when we look at aggregate signals, changes in a way similar to statistical physics from single neurons to populations of neurons. And when I say aggregate signal, I'm thinking about things like the local field potential or an fMRI, the bold signal. The idea behind using an aggregate signal is that we reduce the dimensionality drastically from the single neuron situation. So if we had in the order of 10 squared ROIs, the problem becomes something that we can actually deal with. Just to illustrate what I'm talking about, on the left I'm showing a graph with 1,000 nodes and 10% connectivity. And on the right I'm showing these colored, where each color indicates that they belong to a specific region of interest or population. I've split that into 10. And now we have, instead of 1,000 nodes, we have 10 nodes. And this, as you can see, is much more of a network that we could use causal inference to solve. So if we can use aggregate signals, this is the way forward to look at the entire brain and deal with this huge confounding problem. However, there's some problems with this approach, at least in concept, and one large one was brought up by Mailer and Cording in 2018, and that is that in statistical physics, generally you're looking at particles or molecules which you can assume have random interactions with one another. However, neurons don't behave in this way. They have fixed connectivity, or connectivity that's changing over time, and it cannot always be assumed that they are interchangeable. So I'm going to explore this a little bit further before going forward. So firstly, there are numerous cell classes in the brain, and each class behaves in a different way and is connected in a different way and is located in a different place. So for instance, there are 28 identified interneurons in mouse cortex when you look at them based on various characteristics such as their transcriptions, their connectivity, or their activity levels. Secondly, activity patterns have been shown to be log normal at various levels. So this is explained in a lot of detail in the Bujaki and Muzuseki 2014 paper. So I won't go into a lot of detail here, but I do recommend looking at that paper. In a nutshell, there seems to be very few highly active neurons and many sparsely active ones. This is not input specific, so that highly active neurons are active in resting conditions as well as in task conditions. So it seems that you just have neurons that are really active and then many neurons that are only once in a while activated. Connectivity also seems to have a log normal distribution, and this has been shown both in terms of anatomy, so spine density, axon caliber, and long range connectivity, which is inferred from track tracing, and function as well, so firing synchrony and evoked EPSPs. There are few cells that fire in synchrony with one another very commonly, but most cells do not do that. So between all of these three types of characteristics, it does seem that the homogeneity assumption is false in the brain to a large extent. But is this a problem? So this may be a controversial slide, and I'm happy to discuss this further in our discussion section. But I propose that maybe it's not a problem, and that there's no need for strict homogeneity in our models. The basis behind this is that we have highly active connected neurons that are relatively few in number, and they appear to play a more general role, so they're active regardless of conditions. And we have other neurons that are active only in specific conditions, and these are subpopulations of interest. So these are subpopulations within the population that we've got an aggregate signal from, and they will be dependent on internal or external conditions. So an inference on an aggregate neural signal might look something like this. Under condition C, a change in the activity of population x1 at time t causes a change in activity of x2 at some time later, t plus dt. So we can put the same inference in due calculus notation. Here I've defined lambda as some function which produces an aggregate signal. And on the left, we're looking at the probability of some aggregate signal in population x2 given a condition C and an intervention where we set x1 to some value zeta. And again, on the right, we have our counterfactual. So we have the aggregate signal in x2 under condition C where we haven't done our intervention. And if we can show that the left side is greater than the right side, then we have support for a causal inference that x1 is causing a change in x2. So what does such an inference imply? Basically that a change, whether manipulated or an intrinsic change, which could act as a sort of internal manipulation, in a subpopulation of x1 is causing a subpopulation of x2 to depolarize and fire action potentials. So notice we're not talking about the entirety of x1, but a subpopulation within x1, and ditto for x2. So importantly, we don't know how many neurons or synapses are involved. We don't know the proportion of principal versus interneurons that are contributing to the aggregate signal. We can approximate this, but it's not clear from which neurons the signal is coming from. We also can't say much about what individual neurons or small ensembles are doing in terms of, for instance, encoding or receptive fields, etc. So this isn't the level of our inference. Instead, we want to support inferences about whole brain effective brain connectivity. So for example, how does information move between populations or subpopulations of neurons across time for a given condition? For me, this is interesting enough a question in its own right to be worth doing. So, importantly, there are a few assumptions that are required to support such inferences, and this is where the real debate might lie. So one of the most important assumptions is that all relevant activity is observable in the aggregate signals. So what I mean by that is that unobserved activity in some third population represents a potential confound. So for instance, if there's a small amount of activity in X3 that is lost in the noise and isn't picked up in the aggregate signal, but it drives, let's say X2, then we may make an improper inference about X1 and X2 because we have not observed X3. So this is a real concern, it's not just theoretical. We know that afferent signals can be amplified. So a small signal that goes into a region can be amplified by recurrent connectivity. So it's not strictly linear. And so it's quite conceivable that the first signal that goes in, the afferent signal, is too small to be detected in the aggregate signal, but the amplified signal can be detected. On the other hand, there's some theoretical evidence that even small amounts of synchronized activity should be observable in the LFP. So this is promising, but it is really something that we need to explore if we want to go further in this sort of approach. A second important assumption is that there's no circularity in the network for the time window of interest. I'm going to just try to show this graphically. So on the left, we have a network with two cycles in it, and the cycle is basically recurrence, which makes it difficult to make a causal inference without ambiguity. But if we split this 3-second window up into 3 separate sub-windows of 1 second, we see that we no longer have a cyclic graph, and we can solve the causal problem much more readily. A third important assumption is that causal relationships are stable under a given condition. So in other words, how consistent is the neural response to a given stimulus or task? There are many internal factors that can bias how the system responds to an external stimulus. One example of that is giving a face stimulus of a famous face that has a differential emotional reaction for different people. This is a large issue as well, needs to be addressed. So to sum up, is it sane to do this? I would argue that it is. Importantly, aggregate signals from populations of neurons allow us to reduce the dimensionality of the human brain by orders of magnitude and to address this confounding problem. The heterogeneity of neurons in a population doesn't have to be a problem, in my opinion, if we accept that aggregate activity at any given time represents subpopulations driven by a certain condition. On the other hand, there are some limitations, including that we cannot extend inferences of this sort to individual neurons or small ensembles, and the phenomena which we infer are limited. It is not clear how generalizable these causal inferences might be beyond specific conditions or even individuals. And these are things that need to be better characterized in the future. Thank you.
Hello everyone. In this talk, we will occupy ourselves with the intersection of biological and artificial neural networks at the level of network topology. We will view this intersection from a comparative standpoint, that is, by considering the brain network of different species. Despite that comparative neuroscience spans more than a century, it is only recently, due to methodological and conceptual reasons, that we can directly compare in a quantitative manner the brain network of different species. A key observation is that brain networks have a characteristic network topology that is not random or all-to-all, and this can be observed across species such as the macaque monkey and the human brain. This key network topology observed in biological neural networks is in stark contrast with the network topology of artificial recurrent neural networks. Artificial recurrent neural networks exhibit a structured network architecture, but a random or all-to-all topology. There is a tremendous renewed interest in the intersection of artificial and biological neural networks due to the impressive advancements that we have all witnessed in the field of AI. The intersection of artificial and biological systems primarily is examined at the level of learning algorithms and stimulus representation, and to a lesser degree with respect to network topology. And that's precisely the focus of the current approach. Let's see some examples that examine the network topology of artificial and biological systems. Here we see neuroscientific work that builds on prior engineering work, where a basic feature that is observed in almost every biological neural network, that is recurrence, is incorporated in a convolutional neural network. By incorporating recurrence in a convolutional neural network, this study reports a better correspondence of the stimulus representations across the artificial system and the different brain regions of the human visual system. Another example has to do with the generation of the wiring of a convolutional neural network based on a small world generator. This experimental approach to create a convolutional neural network with small world topology showcases the potential of incorporating network topologies in artificial systems that are analogous to topological configuration found in brain networks. However, here we have to do with an abstract lens through which we assess the intersection of artificial and biological neural networks. This may be actually an advantageous level of abstraction from an engineering standpoint, but when we have a more neuroscience-driven research agenda, and we need to use artificial neural networks as models for the brain, and test hypotheses around functional consequences of structured network properties, then we need to move beyond analogies and instantiate in artificial systems the actual empirically discerned network topology found in nature. With this motive in mind, we instantiate the empirically discerned brain network topology found in different species in artificial recurrent neural networks. Let's unpack the motivation and goals in more detail. Explicit incorporation of the network topology of biological neural networks in recurrent neural networks, artificial systems, is needed if we want to use these artificial systems as brain models. Moreover, this approach is essential to test postulations that suggest a role of network topology in aspects of innate cognition. For instance, the use of network topology as a structural prior that bestows speed of learning in biological and artificial systems. The goals of the approach are as follows. First, we will examine strategies that are used to construct recurrent neural networks based on empirical data pertaining to brain network topology. Second, we will assess the impact of the incorporation of this empirically discerned network topology in artificial systems on the performance of the system. Here, performance we denote two dimensions: prediction capacity and speed of learning. Third, we will examine the above recurrent neural networks constructed from brain data by using three different sources: macaque monkey data, marmoset monkey data, and human data. Thus, this comparative approach offers universality or specificity of the comparisons between artificial and biological systems. So let's iterate now the first strategy. In the first strategy, we assume that each brain region is inhabited by n neurons. Here, in the example, n equals 4. Subsequently, we connect the neurons in the different brain regions in such a way that we comply with the empirically discerned measurements concerning the region-to-region network strength. Specifically, in this strategy, the sum of the weights of the neuron-to-neuron connectivity between region A and region B equals to 5, since this is what is dictated by empirical data. The key point here is that neuron-to-neuron connection weights have diverse values, with the constraint that the sum to the empirically discerned value in the example 5. Moreover, at the binary level, so what neuron connects to what, we also are constrained by empirical data. So neurons in region A connect to region B neurons, but not C, because that's what empirical data dictate to be the case. As a last step, we map the weights from the empirical data to weights suitable for training artificial recurrent neural networks. Therefore, we map randomly initialized weights, if we have Lie or Hare scheme, for instance, to a rank ordering so that they match the rank ordered weights discerned from empirical observations. Strategy 2. We have exactly the same situation with only one difference. The only difference is that now we have equal weights between the neural populations between region A and B. Hence the name of the strategy, topology weight mapping equal. And everything stays as reiterated before. In the last strategy that we tag as binary no-weight mapping, what matters is, as the name suggests, binary topology, what connects to what, and not the weights. Thus, the network topology of this type of recurrent neural networks is only constrained from empirical observations at the binary level. Therefore, we have three strategies for instantiating empirically discerned network data in artificial systems: topology, weight mapping diverse, topology, weight mapping equal, and binary topology, no weight mapping. We will examine the effect of incorporating empirically discerned network topology in recurrent neural networks in the following modeling setup. We use Elman networks, backpropagation of time as a training algorithm, and we use rate models, so continuous activation functions. And also, we can see in this companion piece that the results that we present here also generalize with echo-state networks. Since our approach has a neuroscience starting point, we examine the performance of the network in a key property that biological and artificial agents should exhibit, that is working memory, the ability to track information across time. Specifically, we have two working memory tasks: sequence memory and back memory. In the sequence memory task, the system has to memorize and repeat a sequence of numbers of length L. In the end-back memory task, the system has to learn to correctly identify if the last number presented in a sequence of L numbers matches the number presented n steps ago, hence the name end-back. Let's see what the results look like. First, with respect to the sequence memory task. For this task and for all species, we do not observe any difference in prediction capacity of the bioinstantiated recurrent neural networks obtained from the three strategies when compared to recurrent neural networks that have a randomized network topology. And just to orient ourselves here in this busy diagram, what is prefixed with R concerns the equivalent randomized counterparts of the bioinstantiated recurrent neural networks. And as we see here, across all species, the minimization of the cost function in the test set is very similar, indistinguishable in all these variants. For the end-back memory, we see that not all strategies for creating bioinstantiated recurrent neural networks lead to the same results. Specifically, the equal weight strategy leads to worsening of performance, as we see here, with respect to all the other strategies, and also with respect to the randomized counterparts of the recurrent neural network. The same picture emerges when we assess the speed of learning of the networks, that is, the epoch in which the best prediction on test data is observed. For the sequence memory task, we observe no differences between the different types of strategies followed and also the bioinstantiated or randomized recurrent neural networks. And this is consistent across species. For the end-back task, again, we observe when we have to do with speed of learning that not all strategies are equal. We observe again a worsening of performance when we instantiate recurrent neural networks with equal weights. So here is a summary of the key points that these results highlight. Not all strategies are equal. Diversity of weights are of importance, and this actually reflects observations at the empirical level from constructions of microconnectors or cell-to-cell connectivity, where you have high diversity of synapses from neuron to neuron. We have seen how we can construct recurrent neural networks with biology-based network topology without sacrificing performance, despite the fact that we don't get better, and of course there are no a priori concrete engineering reasons to believe that this will be the case. And this reflects a recurrent theme in the literature, namely that the challenge is to enhance neurobiological realism while at the same time without sacrificing performance. And at least for now, there are not concrete examples where enhanced neurobiological realism enhances also performance. And also we have seen that in this model setup, network topology is not a useful structural paradigm. And what is important for the future is to consider other dimensions of evaluation beyond prediction, performance, and speed of learning. For instance, the one cost of the systems, or what is the case when we have a setup where transfer learning is the desirable dimension of evaluation. With that, I would like to thank the funding agencies and the collaborators, and also point out that all these tools to execute these experiments and convert empirical observations to ready to be trained artificial neural networks are available for the community to experiment with. Thank you very much.
There seems to be a paradox in neuroscience. Every year, thousands of neuroscientists publish thousands of papers, and lots of these papers have lots of cool results, and yet, despite all this activity, we seem to be making little tangible progress in our understanding of brain function or dysfunction. Thomas Insel, the former director of the US National Institute of Mental Health, made exactly this point when he said, "I spent 13 years at the NIMH really pushing on the neuroscience and genetics of mental disorders. When I look back on that time, I realized that while I think I succeeded in getting lots of really cool papers published by cool scientists at fairly large costs, I think $20 billion US, I don't think we moved the needle in reducing suicide, hospitalizations, or improving recovery for tens of millions of people who have mental illness." So what lies at the nature of this basic disconnect? In this talk, I would like to offer a plausible explanation. Let us first note that the basic aim of science is to better and better approximate reality, to find truer and truer models of reality. Now, of course, the ground truth is unknown and inaccessible, and in this situation, the best we can do is to show that our new model is closer to the truth than some sort of strong rival model. In fact, one can argue that science makes progress when it finds truer and truer models relative to stronger and stronger rivals. This process resembles closely the natural selection of fitter and fitter life forms. Now, how does this process work in practice in network neuroscience, the focus of the present talk? So in network neuroscience, we typically start with a network. We represent it as a matrix, we analyze it, we compute some results, and then we often interpret this result in the context of brain function. And this is the first key step in our analysis, but of course it is not the complete analysis, and it must be followed by an even more important second step, where we then compare our result to a rival explanation, an explanation most commonly defined by a null model. Now, in practice, null models define data distributions, and sampling from these distributions allow us to generate surrogate data. Analysis of the surrogate data generates a distribution of null results, and then the comparison of our new result against this null distribution allows us to then accept or reject our null hypothesis. So this constitutes the basic pipeline of analysis in network neuroscience. And in this pipeline, the key question is, which null model are we going to use to test our new result? And in the following slide, I would like to distinguish between two types of null models, which I call benchmarks and strawmen. So first, let's talk about benchmark models. Benchmark models, or well-specified models, are models that are informed by all assumptions of existing knowledge relevant to our question of interest. These models in practice include features that map to biological traits that we think are causally or mechanistically associated with function or development relevant to our question of interest. It is key to compare our new results against benchmarks in order to rigorously test the novelty of these results. But that's not what happens in practice. And in practice, what happens is that we test results against strawman null models. Strawman null models, or underspecified null models, which are informed by few assumptions of existing knowledge relevant to our question of interest. And the tests of our new results against the strawman, in fact, do not constitute rigorous tests of novelty, and strawmen are easy to reject, but such rejections do not really tell us whether we have discovered anything new, whether we have advanced our understanding of brain function. Moreover, in many cases, existing knowledge, assumptions of existing knowledge that are typically specified in benchmark models, can predetermine our new results. And in such cases, our assumptions in effect predetermine our conclusions, and our result is circular. We can also say that our new model is overspecified because it contains a redundant explanation. Now, network neuroscience has many results, and the number of results in the literature continues to grow. And there's really no time in this talk to discuss all of these results, and it's probably unnecessary. So instead, what I would like to do in the next couple of slides is to illustrate what can go wrong when we test the novelty of these results against strawman null models. Now, I will do this using a couple of results that I think represent some of the best known achievements of the field over the last decade. And these results focus on the concepts of the core and of controllability. Let's first consider our toy example dataset on the left. You can see that this dataset has some structure, it has some module structure, it has some gradient structure, and a controllability analysis of this dataset with the result shown on the right suggests that it may contain a structure known as the high control core. Now, the high control core, delineated by dashed lines, includes a group of nodes whose simultaneous activity may be induced with little input energy, and we may hypothesize that the simultaneous activity of all nodes within the high control core constitutes some kind of default state of brain activity. This is an interesting new hypothesis, but in order to show that this hypothesis is significant, we must, of course, test it against alternative explanations. Let's first test it against a benchmark model informed by all key assumptions of existing knowledge relevant to the question of interest. Now, in this case, we informed our benchmark by the observed module structure and gradient structure that is apparent in the empirical dataset. The controllability analysis on samples from the data distribution defined by this model allows us to find a presence of a very similar looking high control core in this model. Okay, and we can quantify this effect precisely using some measure of high control corners, and the effect sizes illustrated on the right suggest that the average effect of comparison of empirical to null hovers around 0, which implies that we cannot reject the null hypothesis, and we must in fact reject the research hypothesis of the additional independence functionality of this high control core. Okay. Here's what happens in contrast when we compare our new result against a Stroman null model. In this case, the Stroman null model is informed by gradient structure, but it is not informed by any other structure. And as you can see on the left, the modules here are missing. Now, the same controllability analysis of samples from a distribution defined by the Stroman null model does not find a high control core, and as you can see on the right, the effect sizes hover well above 0, which implies that we may reject this null hypothesis, and in this case accept our research hypothesis. However, as we already observed, comparisons against Stroman null models tell us very little about actual novelty or significance of our result. Moreover, we have already shown that assumptions of existing knowledge predetermine this result, and in this case, our assumptions predetermine our conclusions. The notion of high control core is redundant, and our ultimate analysis is circular. So returning to the paradox I posited at the beginning of the talk, well, I think it is not really a paradox when we consider the fact that most analysis in the literature, at least in the network neuroscience literature, are based on rejection of Stroman null models. And in this case, the massive redundancy that arises as a result of this analysis allows us to explain the disconnect between the continuous seeming rapid progress that we observe in the literature and the lack of genuine new understanding about brain function that we observe on longer timescales. This is an important problem, and we must urgently do something about it in order to place the field on a rigorous foundation. In the remainder of this talk, I would like to discuss some potential solutions that I hope will allow investigators to overcome this problem and in this way to make genuine progress in this field. The solutions I'm going to discuss are fairly simple conceptually, but they may be quite hard to implement in practice. Now, the first solution should center on a definition of benchmarks. I have stressed this point a couple of times before, and I'm going to stress it again. In order to claim that we are learning something new, we must first understand what we already know about the question of interest. The definition of benchmarks could be done in several different ways, and here I'm going to describe one framework that I think is particularly useful for unifying knowledge and for allowing us to build these increasingly strong benchmarks informed by this knowledge. This framework is due to Tinbergen, who first formulated it almost 50 years ago in the context of animal behavior, but the framework is much more general and it can be applied to the study of any biological trait. Most generally, it asks 4 questions or levels of analysis related to the function, structure, development, and evolution of any trait. These questions could in turn be heuristically subdivided into 2 axes. The first axis distinguishes between the nature, the structure and function of the trait, and the origin, the development of evolution of the trait. The second axis subdivides the timescales at which these questions could be investigated, the immediate timescale of a single life and the historical timescale of many generations. In the context of network neurosciences, we could posit what we already know about specific behavior and the traits that give rise to the behavior, the structure of this trait, and the developmental mechanism that gives rise to the structure. And together, all these features should be incorporated in our benchmark models. Second, once we have our benchmark models, we must then sample data from the distribution defined by these models in order to rigorously test the novelty of our new result. In practice, the sampling from richly structured distributions can often be slow and hard and may not scale to large datasets. And so the development of methods that allow us to perform such sampling without bias, i.e. without any additional assumptions, would be an important step that would place the field on a more rigorous foundation. My lab has been working in this direction, and here is a project that we've been recently working on with many collaborators, where we have developed methods that allow us to sample large neuroimaging and neurophysiological datasets with increasingly many spatial and temporal constraints. So here, a few constraints are illustrated, which include constraints on the individual activity of every node in our data set, as well as at every time point, and the correlation between individual nodes, groups of these nodes, which may be defined as systems or modules, groups of time points, as well as spatio-temporal modes. Here's an example application of these tools to the study of questions in human neuroimaging. So let us first look at the column on the right. It shows the data, and you can see that this data has a map of the default network. And so what we sought to do is we sought to explain the extent to which this map could arise through constraints on interactions between other networks in the system. And we considered other cognitive networks that do not include the default network. First, we started with no network constraints at all. This is our Stroman null model. On the left, you can see there's very little structure in this model. But then, interestingly, as we start to add more constraints, in the second column, when we have about half of all network correlations, we find that we see a map that starts to resemble the default network in many of its key elements. Not all, but many of its key elements. And then when we constrained all possible network interactions, we of course see that this map becomes more and more refined and start to resemble the structure of the default network more and more accurately. We have then continued and performed a similar analysis to test the dynamic fluctuations of individual voxels against the static maps. So the data on the right shows voxels that most strongly fluctuate in their correlation maps against their static maps. And you can see that the voxels that most strongly vary in their correlations, again, center on parietal, temporal, and to some extent frontal association regions. And when we looked at our three models from the previous slide, again we observe that as we start to add network interactions, we start to pull out some of the same areas associated with high variance in correlation maps. Note importantly that we did not constrain here any dynamic interactions. We simply constrained the static correlations between networks. So in conclusion, the problem I delineated here is important and requires an urgent solution. And the solutions that I have outlined are fairly common sense and involve taking stock of all existing knowledge, testing new results against benchmarks, in this way minimizing redundancy. And I hope that collectively this process will allow the field to make genuine progress and in this way get closer to truth.